questions, you can also follow us to Zoom. And we will add this link in the chat later. So now I'm happy to briefly introduce our speaker, Dr. Kate Kuhlman. Kate is an assistant professor at the University of California, Irvine. She received her PhD degree from the University of Michigan in 2014, and she then moved to UCLA for postdoctoral training. And Kate is the director of the UCI Teen Resilience Lab, and her research focuses on how early life adversity affects health. And today she will present results from the recently completed Teen Resilience Project. So thank you so much for being here with us today, Kate, and for sharing your interesting work. So now we all look forward to just sit back and to listen to your talk.
Thank you so much, Lena. Thank you for having me. I'm excited to be here. So today I'm going to talk to you about a project that kind of was the epicenter of my overarching program of work, which focuses on the role of psychological stress in risk for depression. What we know is that psychological stress can affect and activate a number of different physiological stress response systems, and that those systems and the molecules that result from their activation can lead to the symptoms of depression. And that over time, this can become an insidious negative feedback loop such that depression can then become a psychological stressor in and of itself, and that this plays out over the course of the lifespan in quite a repeated and chronic and persistent way. And for that reason, a lot of the work that I do centers on this main thesis that if we can identify modifiable factors that reduce risk for the onset of this insidious pathway, particularly among young people who've been exposed to a lot of adversity in childhood or a lot of psychological stress, we might be able to substantially reduce the burden of depression on society. So I'll focus on a little bit of background for why this is really what I've decided to spend my kind of academic time studying. First, by introducing why depression is really the outcome. We know that adversity and stress has been linked to a number of different physical and psychiatric disorders. So why focus on depression? We know that depression is quite prevalent. So in the United States, about 10% almost of the population will experience a depressive episode anytime in a given year, and at least 30% of the population will experience this disease at some point in their life. We also know that it's persistent. So once a person experiences a depressive episode, they have at least a 50% chance of having that disease recur, even if they've been treated effectively. We also know that it's costly. It costs a lot of healthcare dollars, but also a lot of suffering and lives lost. And this is a lot of global data as well. And we also know that it's getting worse. So in the 20 years between 1990 and 2010 in the United States, we saw almost a 40% increase in the prevalence and the cost associated with this disease. And that we have only reason to believe that those numbers in the subsequent 20 years are going to get worse. So why adversity? So I like to pause here and just kind of introduce this idea that early life adversity can be defined in a lot of different ways. And a lot of people use very specific definitions of early life adversity, things like childhood maltreatment, things like being exposed to very specific traumatic experiences in childhood. I take a broader definition that's much more consistent with the adverse childhood events model that was developed by the Center for Disease Control here in the United States and Kaiser Permanente, where I think of it as non-normative experiences during development, things that are uncommon, that have the potential to alter developmental trajectories at some level of analysis. If we take that definition and we use that Kaiser and CDC data, what we know is that about a third of the adult population in the United States is exposed to no early life adversity by this definition, but about 12% of the population is exposed to 4 or more. And this population exposed to 4 or more are at very grave risk for a number of different physical and psychiatric conditions. There's a dose-response relationship. So if you have one additional early life adversity exposure, you have one, increasing risk for a number of different diseases. In this high adversity exposure group, we have earlier mortality, double the risk for heart disease, cancer, and stroke, 4-fold increases in depression, and also a 12-fold increase in suicidality. So you're seeing this really substantially increased risk for these diseases. And I also focus on doing most of my work during the adolescent developmental period. And the reason for that is that we know that it's a sensitive period of development from a social, neural, and cognitive perspective, which means that being ill or having a depressive episode during that period of time can have compounding impacts on our development over time. But also being well during that period can have compounding positive impacts on our development over time. And so if we can leverage treatments during this period of time to promote wellness, we might be able to reduce these long-term negative outcomes. And just as a proof of principle, this is just some recent data that my group published with a really phenomenal student, Kathy Chang, where we used the National Comorbidity Survey replication data to just ask this question as a proof of principle. So among the 3,360 people in the sample who had ever experienced a depressive episode before, we compared adulthood social functioning between the individuals who had a depressive episode that began in adolescence or a depressive episode that began later in adulthood, or age 21 or older. And what we see is that there's a substantial increased risk in some of these non-normative social outcomes in adulthood. And some of these people were followed in the sample or assessed in the sample in their mid-40s. And so we have good reason to believe that there are kind of long-term social consequences of being exposed to depression during adolescence specifically. And this was controlling for the number of weeks a person was depressed across their lifespan, and things like recurrence and stuff like that. So for this reason, we really like to focus on adolescence and what we can do to leverage well-being during that adolescence. Other reasons to focus on adolescence have to do with the high rates of suicidal ideation among depressed adolescents. There's a much higher risk of suicidal ideation and behavior. And we also know that in most countries and a lot of the United States, you have an increasing amount of autonomy in adolescence, where individuals are legally allowed to consent to mental health services during this developmental period, allowing them to then gain cognitive behavioral skills and other skills therapeutically that can help promote well-being and resilience. So hopefully I've given you a little bit of food for thought about why this is so important and why you would want to focus on depression and kids specifically with early adversity exposure and try to understand how to leverage these things during adolescence. The modifiable pathway that I spend a lot of time thinking about and studying in my lab is the neuroimmune network, the neuroimmune communication or network, which is really trying to understand how the immune system engages the brain in an upstream pathway. So how inflammatory cytokines and myeloid lineage cells in the immune system affect the brain and behavior in ways that either can promote resilience or can lead to symptoms of depression or increased risk. What we know is that if an individual, or in this case, Fido here, eats something or consumes something that's pathogenic, that their immune system engages activates and starts to produce cytokines. Inflammatory cytokines then traffic to the brain or influence the brain in a way that increases your thermoregulatory set point, mounting a fever, and also changes behavior in a lot of ways. So increases your slow wave sleep, decreases your appetite and your exploration of the environment in ways that hopefully keep you from eating whatever pathogen you ate in that environment before. But these constellation of changes over time lead to our ability to recover from that pathogen and become well again. What we know from a number of different advances in the field, so this cartoon is from 1988, we've known this for a very long time, but more recently, we've been able to characterize the neural underpinnings of these processes, knowing that inflammation can change dopaminergic signaling, decrease neurotrophic factors like BDNF, and decrease serotonin, while also simultaneously increasing excitotoxicity in the brain via glutamate. So we've mapped out a lot of we very carefully mapped out a lot of different important pathways through which inflammation can lead to all of these changes in behavior. And during my postdoc, what we spent a lot of time focusing on was, the extent to which these symptoms overlap with depression. And so we did this kind of scoping review where we identified all of the different ways that you can experimentally activate the immune system in a human model. So things like giving vaccines, administering LPS or endotoxin in the lab, or some of the clinical trials that use interferon that activate the immune system. We kind of looked at all of the different endophenotypes that we see in depression, so kind of clusters of symptoms that have kind of a heritable shared underpinning, and saw that no matter how you influence the immune system, how high of a dose of immune activation you give a participant, you get changes to somatic symptoms like fatigue and changes in sleep and psychomotor slowing. At higher doses of immune activation, you get changes to reward reactivity or reward processing more broadly. At lower doses or more acute activation of the immune system, you get negative affective effects. So kind of more negative affect responses to stressors in the lab, more negative affective responses to kind of threaten the environment, which gave us, a lot of reason to believe that, inflammation can play this causal role in the development of at least some of the major and impairing symptoms of depression. And so it's not surprising then that we've seen across the literature over the past 2 decades quite a lot of evidence that individuals with depression relative to individuals without depression tend to have slightly elevated circulating markers of inflammation. So here you see in adults with depression, C-reactive protein and interleukin-6 are significantly elevated among individuals with depression. We also see some evidence that in some clinical samples that these markers also prospectively predict the onset of depression. So this is just some kind of example data in one clinical sample with diabetes, where individuals with high levels of circulating C-reactive protein were more likely to develop depressive episodes over time. And a lot of my work has then tried to superimpose our understanding of how early life adversity contributes to these pathways. So if we have pretty good evidence that inflammation can lead to depressive symptoms, how is early life adversity affecting this? And a lot of people think or have, tested the hypothesis that early adversity can increase the pro-inflammatory phenotype of an individual, resulting in their body mounting a larger inflammatory response to being in some sort of immune challenge. So any of those immune challenges or a stressor in your life. And we see that in adults, that individuals exposed to more early life adversity tend to have higher circulating C-reactive protein, higher interleukin-6, and higher TNF-alpha. We did this analysis, this meta-analysis in pediatric populations. There are far, far fewer studies in this field, but we had sufficient data points that we could do the meta-analysis for C-reactive protein and interleukin-6, and saw evidence that C-reactive protein was indeed significantly, but not very robustly associated with, or that early life adversity was significantly, but not very robustly associated with C-reactive protein elevations. And there was less consistent evidence for interleukin-6 that might be developmental in nature. So if we have more studies, we might be able to interrogate that further. We also have seen evidence that individuals with a exposure to specific forms of early life adversity, so in this case, this is individuals with maltreatment exposure during childhood, mount a larger inflammatory response to stress in the laboratory. But this has only been done among adults. We also have evidence that early life adversity might affect the regulatory capacity for inflammation. So we know that glucocorticoid circulation can inhibit the production of inflammatory cytokines, but that individuals exposed to early life adversity tend to show an attenuated or blunted glucocorticoid response to acute stressors in the laboratory, suggesting that you have these individuals with early life adversity producing more inflammation, potentially because of a pro-inflammatory phenotype, but also potentially because they have less glucocorticoids being produced simultaneously, and then kind of downregulating the production of inflammation. And taken together, this formulated kind of the first aim of what we call the Teen Resilience Project, which is just to better understand how early life adversity might shape immune responses to acute stresses in the laboratory, and particularly focusing on this in an adolescent population, because as I pointed out throughout, a lot of this work has been done in adults, and a lot of the work, the studies that have been done in pediatric populations are few and far between, despite all of the important reasons why we need to understand these processes during adolescence. So the Teen Resilience Project was part of my Career Development Award from the National Institute of Mental Health. We were really interested in oversampling for individuals exposed to early life adversity, and I feel like that's one of the hardest lessons that I learned throughout the K Award, is like how to do that well and how challenging that can be. So we used census-based mass mailing to invite individuals from specific zip codes around UCLA and UC Irvine. We focused on kids between the ages of 12 and 15. We did that because in the United States, we know that about 4% of 12-year-olds and about 14% of 15-year-olds have depression. So across those 3 years of adolescent development, you see this kind of rapid rise in the prevalence and risk for depression. So we recruited teenagers in this age range, and we excluded anyone who currently or had MDD, or major depression, or had a history of it, so that we could potentially follow these kids while they were developing their first episode. Before the COVID-19 pandemic hit everybody, we had recruited 97 kids into the study, so that ended up being our final sample. They were about 14 years old, a little bit less than half female, and they were, racially and demographically and ethnically matched to the local Southern California area. We used the Adverse Childhood Events questionnaire or as a structured phone interview with parents during the eligibility screen. We asked them about whether their child had ever been exposed to emotional abuse, caregiver mental illness or substance abuse, poverty or bereavement, parent divorce or separation, any housing instability, domestic violence, or caregiver incarceration. And so with this list, the parents could indicate just yes or no, simply whether their child had ever been exposed to any of this. The possible range for exposures then with these 9 items was 0 to 9. And in our sample, we observed a range of 0 to 7, with on average participants exposed to about 2 of these items. And just to kind of underscore the efforts that we had to oversample for early life adversity, I'll remind you that what we know from adults is that about 31% are exposed to none of these, and about 12% are exposed to 4 or more of these. And our sample ended up having about 10% of the sample exposed to none of these, and about almost 20% exposed to 4 or more. So we brought these teenagers into our lab, and we administered the Trier social stress test for children in order to activate the innate inflammatory immune system. We measured their positive and negative affect throughout the entire laboratory protocol to determine whether the TSST was effective the way that it's been seen to be effective in a lot of other labs. Here, I'm showing you the change in negative affect that we observed in the sample from baseline. So basically, you see at 15 minutes post TSST starting, you see this big increase, about a medium to large effect size of negative affect that recovers throughout the rest of the protocol. So we were pretty happy with that. We also inserted an indwelling catheter and collected blood immediately before starting the TSST, 60 minutes and 90 minutes later. That enabled us to characterize the innate immune response to the stressor. We did this at multiple levels of analysis. So we looked at C-reactive protein and then a panel of cytokines via multiplex in plasma. We isolated RNA pellets so that we could look at inflammatory and antiviral gene expression changes. And then we also use flow cytometry to characterize changes in classical, intermediate, and non-classical myeloid lineage cells, specifically monocytes. One of the main findings of the study was that we saw exaggerated immune activation in these kids exposed to high early life adversity. We saw significant, at least twofold change in expression of immune-related genes from pre to 60 minutes post-stress. It was particularly driven by this high-risk group where their ACEs score, their early life adversity, was around 4 or more. But this is a priori set of about 54 genes that we associate with the immune system and doesn't really give us a lot of resolution about the kind of components of the immune system that are being affected, whether they're innate inflammatory or antiviral in nature. So if you break them down here on the screen, on the left, you can see their inflammatory gene subset. And on the right, you can see their antiviral gene subset. And what you can see is that it's clearly being driven, this exaggerated activation in these kids with exposure to high early life adversity is being driven by these innate inflammatory genes. You see some evidence of this in the antiviral gene subset. I wouldn't say it's absent, but it's much less robust. And it drops to non-significance when you account for important covariates that we like to account for in inflammatory biology. Consistent with our meta-analysis and also the meta-analysis in adults, we did see this graded kind of dose-response relationship between C-reactive protein and early life adversity, where kids with early life adversity in this highest 4 or more range had the highest circulating C-reactive protein concentrations that declined with lower early adversity in the rest of the sample. But we didn't see any evidence that that circulating markers mounted a larger response. So here you can see the black dotted line are the kids exposed to no early life adversities on our checklist. And they show kind of this fast, robust response when measured by IL-6 in response to the stressor. They kind of meets a peak at around 60 minutes post-stress and then plateaus or starts to recover even by 90 minutes. By contrast, the kids exposed to early life adversity, both high and kind of in the middle, show this attenuated increase in IL-6 at 60 minutes post-stress. But by 90 minutes post-stress, their IL-6 concentrations aren't significantly different from the no exposure group. And one of my kind of future hopes is that I'll get to do this study again and have a longer term follow-up, like at 120 or 180 minutes post-stress, because it's quite possible that this pattern might show that these kids exposed to high early life adversity might be continuing to show increases in inflammatory markers, circulating inflammatory markers, whereas these kids with little to no adversity exposure might decline over time and be recovered by those time points. So in the future, maybe I can come back in a few years and give that talk about that data. And this is just that same data presented in a different way in terms of kind of delta interleukin 6, where you can see that at 90 minutes, these kids with high early life adversity are kind of starting to catch up with the kids who mounted a robust response at 60 minutes. So overall, our first aim of the Teen Resilience Project was quite enlightening. So we saw some evidence that our data mapped on to what the larger epidemiological evidence would suggest, that early life adversity was associated with higher systemic inflammation as measured by C-reactive protein. They mounted a larger inflammatory gene expression response to the stressor, but we didn't see the same pattern that you see in the adults exposed to maltreatment with respect to these circulating inflammatory responses to the stressor, though that may be that they had a protracted response that we'll have to investigate further later. But this pattern of results doesn't really seem compelling enough to kind of explain why we see this 2 to 4-fold increase in depression and these other inflammatory disorders that we see in these kids with high adversity exposure. And so I've been very interested in whether there's more to this story. Robin Nusslock and Greg Miller have kind of a driving theory called the neuroimmune network hypothesis that suggests that early life adversity does many things to the neuroimmune system. So, what we just talked about is on the bottom left-hand side of the screen, showing that early life adversity kind of promotes this pro-inflammatory phenotype among monocytes and macrophages, leading to kind of chronic low-grade systemic inflammation. But that early life adversity also affects the central nervous system more directly, changing threat and reward sensitivity, and particularly being affected by kind of activated or primed microglia that further perpetuates these kind of changes in threat and reward systems. And in particular, what they argue is that kids exposed to early life adversity might be more sensitive to these pathways. So they might be producing more or having more activated or pro-inflammatory microglia, leading to a more robust effect of inflammatory signaling on these systems. And this is a question that I've been interested in for a while, and something that we have found in other data sets. So just really briefly, we did during my postdoc, we did the UCLA flu vaccine study, where we were interested in whether the flu vaccine could be used as one of these exogenous challenges to the immune system, the way that other inflammatory challenges have been used, like endotoxin or the typhoid vaccine. And so we kind of characterized the time course of response to the immune system, or response to the flu vaccine, where the vast majority of people show a peak inflammatory response about 24 hours after receiving the vaccine. Then we did a daily diary study where we brought people in and we got a really good 7 days of kind of daily mood variability. We collected blood before giving them the flu vaccine, brought them in 24 hours later, collected blood again for characterizing that peak immune response. And then we followed them for another week to get changes in mood across the rest of the week, with the idea that, if this works as a similar immune challenge, we would see that the inflammatory response tracks with the affective or mood response that we see. I think in the interest of time, so this was just an undergraduate sample, so about freshmen at UCLA, very healthy sample, and looked very similar to all of the, kind of the demographics of UCLA. We did see a lot of variability in the way that people responded to the immune system or responded to the flu vaccine. This red line on the screen shows the average change. So really small, modest, kind of mild change in inflammation, but reliable and significant. And the black lines show anyone who showed an increase at all, and the gray lines showed anyone with a decrease. So we saw most people showing some sort of increase, but it was mild for most people. But that being said, the variability in their IL-6 response then did track with changes in depressed mood and changes in cognitive, like subjective cognitive symptoms. In this case, it was the POMS confusion subscale, where individuals who showed the largest change or increase in IL-6 from pre to post-vaccine also showed the largest increase in both of these symptom domains. And I'll just note that this one person who looks like a big outlier our effects actually grow more robust when you remove that person. So in the parentheses are the coefficient estimates if you remove that person who showed a much bigger change in IL-6 than everyone else. And what you can see is that the time course is really interesting, that across, so this day 0 is the day that they received their flu vaccine, and that individuals who show a larger IL-6 increase tend to show the biggest differences in their mood ratings at 2 and 3 days post-vaccine exposure. So kind of the mood symptoms seem to be at least temporarily following the change in inflammation that we're measuring, or that peak that we identified. But we also measured exposure to early life adversity in these kids, or in these undergraduates. We asked them a number of 50 different potentially traumatic experiences that cross physical, emotional, and sexual trauma. And we asked them how many times it's happened and how old they were at the time. And overall, what we found was that these effects of, change in IL-6 tracking with mood, as well as changing IL-6 tracking with confusion or subjective cognitive problems, was really driven by the kids exposed to the most early life stress. So here in the black line, you see the kids exposed to high levels of early life stress. So about six events on this traumatic stress checklist relative to the kids exposed to little to none. And you see the same exact pattern with cognitive difficulty or confusion, suggesting that the neuroimmune network hypothesis is quite, that there's growing evidence in support of this idea that these kids with early life adversity might be more sensitive to the inflammatory, the behavioral consequences of inflammatory signaling. And in the Teen Resilience Project, we really wrote this grant in order to run the study in an adolescent sample and ask these questions in the context of adolescence and stress in the laboratory. So to do that, the same protocol that we saw before, but upon arrival to the lab, they did a series of behavioral tasks that measure reward and risk processes. And then between 60 and 90 minutes post-stress, we administered those tasks again. And the tasks that we used were the balloon analog risk tasks, because the neuroimmune network hypothesis does focus quite a bit on kind of risk-taking, kind of poor health behavior model. And then also the pirate task, which is a reward learning task. So in the BART task, participants see a red balloon on the screen, and they're told that they can pump the balloon up as much as they would like. And every time they pump the balloon, they get 5 cents. That they will then receive at the end of the experiment. They're then told that balloons pop if you pump them up too much, and if their balloon pops, they lose all the money that they've earned on that specific balloon. So if at any point they don't want to risk losing that money, they can click collect your money, and all of the money they've earned on that balloon will be added to their overall bank. And then they'll move on to the next balloon, and there are 30 balloons in total. And so the outcomes in this task are really just how many times did they pump up the balloon overall and per trial. And what we saw is that from pre to post stress, kids became more risk oriented or more reward oriented. They pumped the balloon up 80, or on average about 80 more times in the post stress administration of the task. But IL-6 changes in response to the stressor weren't related to this behavior change at all, pre and post stress. We did, however, find that among the kids exposed to early life adversity, that was a significant moderator of the role of IL-6. So I'll show you what that looks like. So among the kids exposed to low early life adversity, you see that having greater IL-6 reactivity was associated with this increase in pumping behavior, or increase in risk orientation or willingness to take on risk to earn more money. That pattern was reversed among the kids exposed to high early life adversity. So the kids who didn't mount a large inflammatory response tended to produce or engage in more pumping behavior, even more substantial increases in pumping behavior, in fact, than the low adversity kids. But among the kids who mounted a large inflammatory response, you see a completely flat pattern where they didn't change their reward motivation or like risk-oriented behavior at all. We also administered the PIRATE task, which is a reward learning task. It's pretty simple. I really like this task because it was developed by a neuroscientist at UCLA named Adriana Galvan, who's in the psychology department. And she developed it to do translational studies where this task can be used with non-human primates and also with human adolescents. And the task just involves seeing a series of pirates on the screen. So one at a time, you see a pirate. They appear either on the left or the right-hand side of the screen. Then there's a 2-second delay and a cue, in this case, treasure chests. When they see the cue, they're asked to indicate whether the last pirate they saw was on the left or the right-hand side of the screen. And then they receive some sort of reward for correct trials. Each participant is assigned 3 pirates, and each of those pirates is assigned a different reward value. So one pirate is associated with earning $1, essentially. One pirate is associated with earning 50 cents or half of $1, and one is associated with earning 10 cents or 10% of $1. And so over time, the question is really whether there's a response bias developed where they are more accurate or faster at responding to the high value relative to the low or medium value pirates. What we see is that the participants become a lot less accurate. So in some ways, this means that the data actually shakes out that they just stop kind of responding to the low value pirate. And so they're getting, being accurate with the high value pirate over time. And we saw that IL-6 was related to their reaction time. So the more IL-6 an individual in the sample produced in response to the stressor, the greater decreases or reduction in response bias they produced at the post-stress administration of the task. So they were not as oriented towards this. There was no behavioral evidence, I guess, or less behavioral evidence that they were oriented towards the high value relative to the low value pirate. And this was also moderated by early life adversity. So in the kids with low early life adversity, when they mounted a robust inflammatory response to the stressor, you again see this like big increase in response bias where they are much more accurate with these high value trials relative to the low value trials. But among the kids with high early life adversity, you see that effect attenuated. So you don't see this facilitating effect for reward learning that comes from the inflammatory response among these kids. And if anything, you see kind of the opposite where they're declining in reward orientation or response bias over time. So this felt like pretty good evidence that the neuroimmune network hypothesis was kind of panning out in our data, that these kids are exposed to high early life adversity might be differentially sensitive to the effect of inflammation on their behavior, which is kind of exciting to us. And one of the overall goals of the study was to try to characterize this. And we're seeing kind of interesting differential patterns, particularly based on the type of reward task that individuals were participating in, whether it's kind of risk orientation or reward related learning, and then some converging paths as well. So the last aim of the study was really to understand whether anything that we were measuring in the lab with respect to neuroimmune processes was related to which kids ended up developing depression. So if you'll remember, we recruited high-risk kids because they were exposed to higher levels of early life adversity, but they hadn't had any depressive episodes in the past and weren't depressed when we recruited them. And then we followed them for 12 months. So the laboratory protocol that I've been talking to you about this whole time was situated within a prospective longitudinal study design, where after they left the lab, we followed them every 4 months for a year. And at the end of a year, we assessed for the presence of any depressive episodes during that period of time. About 17.5% of the sample developed a depressive episode over the 12-month follow-up. This was much higher than we were expecting. And if you remember, we had to shut down in-person data collection right when the pandemic happened. And so there's a little bit of evidence that the rates of depression development during that first year of the pandemic were a little bit higher than the rest of the sample, but not so much. I certainly don't think the pandemic helped. And we did see that this development of depression did track pretty well with the early life adversity exposure, such that the kids in the sample with no early life adversity exposure, none of them developed depression. And the kids in the sample with 4 or more, so that high-risk group I told you about 20% of them developed depression over the 12-month follow-up period. This seemed to be particularly tied to symptoms of anhedonia. So we use the Reynolds Adolescent Depression Scale to measure depressive symptoms via self-report at each of these 4-month follow-ups. And what we see is that these kids with higher early life adversity tend to show this kind of trajectory of increasing anhedonia symptoms over time, where these kids with no early life adversity or little to no early life adversity kind of show a flat or even declining trajectory of anhedonia across the 12 months that we followed them. Unfortunately, none of our inflammatory markers were related to depression onset. So we didn't see any significant associations between any of our circulating inflammatory markers and depression onset. Here in blue, you see the kids who went on to develop depression, and the kids, the black bars indicate the kids who went on to or didn't develop depression over the follow-up. The only kind of marginal or trending association we observed was really for interleukin 10, which is kind of a canonical anti-inflammatory cytokine. What we see here is that the kids with higher interleukin 10 concentrations at baseline tended to be at higher risk for developing depression over time. And the biggest predictor of developing depression that we observed in our sample, or at least that we've kind of identified so far, is executive functioning problems. We assessed executive functioning problems in this case via parent report on a clinically validated instrument of behavioral symptoms called the brief. And the brief has 3 different composite scales, so a behavior regulation index, as well as a cognitive regulation index and an emotion regulation index. I think what you can see by just eyeballing this figure is that in the black bars here now, the kids exposed to early life, or the kids who went on to develop depression, just tend to have higher symptoms or higher T-scores in all of these domains. But the kids who don't develop depression have, lower symptoms in all of these domains. But the biggest effects really come from the emotion regulation executive functioning domain. So you see it for the overall emotion regulation index, you see it for the shifting abilities, and you see it for emotional control. And then we also saw a trend with organization of materials, but that falls outside of the emotion regulation domain. So executive functioning skills, particularly in emotionally valenced contexts, really seem to be what are differentiating these kids based on their parents' report for who will develop depression within the next 12 months. And so this caused us to kind of interrogate, whether inflammation was related to any of these. So do you see any evidence that this kind of trend, non-significant association between IL-10 and 12-month depression onset might be explained in part by the effect that interleukin 10 could have, or that inflammatory signaling more broadly has on executive functioning domains? So we conducted a parallel mediation with all 3 of these executive functioning domains as competing mediators. And what we see is that interleukin 10 was significantly associated with having more problems in this emotion regulation domain of executive functioning, and that this emotion regulation domain of executive functioning, as we saw, was significantly related to which kids 12 months later would have developed a depressive episode. And this is really interesting. Ariel Radin is a colleague of mine at UCLA, and she's very interested in the role of inflammatory signaling in executive functioning broadly and cognitive functioning. And so I've heard a lot and heard her talk a lot about this. But I'm really interested in early life adversity. And so I was really interested in the way that early life adversity might be contributing to these patterns. And so I looked at whether there was a moderated mediation here, such that this like role of IL-10 in this executive functioning domain was varied by early life adversity background. And what we saw was that it was really driven by the kids exposed to some early life adversity much more robustly among the kids exposed to higher early life adversity. So, having high IL-10 was really related to deficits in emotion regulation, domains of executive functioning among the kids with high or at least average. So this is 2 early life adversities or more than kids with low exposure. And that explained this entire direct effect between IL-10 and 12-month onset. Of course, this main effect was non-significant. It's only 97 people. And so we're really putting a lot of strain on this model, but I think it's really exciting and an interesting possibility that we're going to follow up on in other data sets. So overall, it seems like what we're learning from this teen resilience project is that these executive functioning deficits might increase risk for depression over time, particularly among adversity exposed adolescents. What I think is really interesting is that I told you about this scoping review that we did a few years ago, looking at all the different ways that you can exogenously activate the immune system. And deficits in cognitive control was the one endophenotype that we really didn't see a lot of really strong evidence for. And that was particularly because, a lot of the studies in that domain used neuropsychological tests that were cold cognitive tests in nature. So they were measuring, working memory and complex figure and shifting and just kind of cold non-emotional context of neuropsychological abilities. And we have a lot less evidence of kind of how these neurocognitive tasks would work, or like how performance would differ under emotionally valenced or stressful contexts, which is kind of the context in which we did a lot of the work that we did in this study, and is an interesting kind of potential future direction that we'll explore more. And so, one of the hopes that we have for this study is that it will breed more funding for us to be able to do psychosocial interventions, particularly finding interventions that have an evidence base for efficacy for depression prevention or depression treatment that might affect executive functioning in emotional contexts. And whether that might disproportionately benefit these kids exposed to early life adversity and kind of mitigate this neuroimmune risk pathway that we've been talking about. So with that, I'll just stop and thank my collaborators on this project, who've been my mentors and cheerleading team and champions over this course of running the Teen Resilience Project, as well as the organizations that funded the work that I presented to you today. And if you're interested in any of the things that my lab does, we have a website here, teenresilience.org. And then also, as of a week ago, we created a Twitter account. And so you can find us on Twitter at teenresilience. And thank you for your time.
Great. Thank you so much, Kate, for your amazing talk. It was so nice to listen, and I learned a lot. So I will remind the audience that you can ask questions with the Ask a Question button. And if it's okay, I would like to start with a small question. So as I'm curious about how you selected the stress test, do you think that you would have different results if you used another type of test and for example a test that's non-social?
It's a good question. I have seen, so I think that I've used a lot of different tasks in the past for different studies, stress tasks in particular. I don't like the TSST because it's long. I do like that it is social. I've used some, I think what we know is that social stress particularly is relevant for the immune system. And so I think it would be an interesting question to kind of pit a similarly timed question or similarly timed stressor in the laboratory that was absent of this social component. But I think what we've learned, at least in the psychoneuroendocrine world, is that having social tasks is more robust. Like they work a lot better than kind of pain or frustration or cold water and things like that. And so I think in these types of samples, what we wanted to do was use what we thought would be the most robust activator. The last thing you want is to run a study like this with so much money and so much effort and time, and then have it not activate the systems that you're interested in, and then not know how to interpret the data. So I think the social component is really important, and I'm particularly interested in that pathway. But I think it would be interesting to kind of do that experiment of randomizing people to a social or non-social version of it to see whether that differentially affects the immune system or its consequences.
Great, thank you for the explanation. We will continue with a question from Yuji. For the phone interview with the parents, would the abusive parents hide the maltreatment? Is that not a bias to take into account? Thank you.
Yeah, so that's a great question. And we struggled with this a lot in our study design of what the best approach to do. So we had parent-reported early life adversity because we wanted to make sure that we would only recruit kids with kind of oversampling for early adversity. So we needed to know something about their adversity exposure before we enrolled them in the study. Once all of the kids enrolled in the study, we then had child self-reports of early life adversity as well. And that allowed us to do kind of convergent validity analyses where the relationship between parent-reported and child-reported early life adversity was pretty concordant. I mean, the correlations and the kind of the group membership, if you put them in these like no adversity, middle adversity, and high adversity groups, those groupings were almost identical. So I think that the parent-reported did give us a good measure. It's also important to note that we left out in the parent interview, we left out child physical and sexual abuse, which is usually part of the ACEs, because at the stage of the eligibility phone interview, we don't really have enough information to be able to make mandated reports about child abuse. So we would be our hands would be kind of tied. We would like learn this information about a child who's at risk and not be able to do anything about it. Once they're in the study, we can do more about it. And so we left those items out. And so hopefully parents who would have said yes or minimized their answers to those questions weren't put in that situation where we asked them, because we didn't ask them those questions. But we were happy to. And the concordance data is published in our paper where we report the immune activation. So we have a supplementary table in that paper that people can look at the way that the parent and the child reported adversity exposure did and didn't match up with each other. I think the other thing that a lot of people don't realize about early adversity measurements is that it is the case that parents minimize things that would have occurred in which they played kind of a causal role, and parents would minimize some adversity. But children also aren't aware of certain types of adversity that happened when they were very young. And so there's kind of error in reporting in both directions for child and parent, which is why in all of my studies I try to get parent and child report just to try to get the clearest picture I can about their exposures.
Great. So we also have a question from Corinna. Thank you for a great talk. Did I get it correctly that the balloon task was used as a measure of reward sensitivity? How are the results affected by effort sensitivity, as participants have to press buttons repeatedly, which can be perceived as effortful? As there is evidence that in depression, effort sensitivity might be altered. Thank you.
Mm-hmm. Yeah, this is a great question, and also something that I've been thinking a lot about lately. I included the BART in the study as a measure of reward motivation, very similar to the way people use the effort task. I used that task because it had a lot of data that it had been used in adolescence. There's a lot of data that you can do it in a repeated measures design like this, doing it once and then doing it kind of 2 hours later in the lab. And so there were advantages. If you look at the meta-analyses of the neural regions that are activated by participating in the effort task or in the BART task, it's very similar to the neural networks that we know are associated with effort. And so I think you're spot on. I think of it that way. What I am realizing is that, depending on the background of the investigator, a lot of people think of this task really just as a risk task. So this idea that, your pumping behavior is constrained by your tolerance of risk. And so higher pumps mean you're willing to take more risks to make more money, and lower pumps are an indicator that you are risk avoidant. And so, I don't think that the BART task is the cleanest effort task or the best measure of reward motivation, but for this study design, it was the closest we could get. But I definitely see it the way that you're describing as more of an effort. I see it as a reward motivation task, and that, higher pumps are an indicator of how much motivation you have to earn rewards, and that occurs in a context of risk, but also other things. But that being said, we didn't see a lot of evidence that people's effort got constrained over time. A lot of the effort tasks that you see in the literature are very long, and they require a lot more effort than just pushing the space bar over and over again. And these kids, they only got more reward motivated over time. They were reporting increases in fatigue, changes in fatigue because they were in the lab for a long time, like over 4 hours. And so they were reporting things like fatigue, but in this task, they only increased the amount of pumping because they wanted to get the money. So I think it would be interesting if you would modify this task to modulate the risk component of it, and maybe use something that wasn't just money, but maybe social rewards or kind of praise, or something that was not so monetary in nature.
Thank you for that great answer. We will have one last question from Rian about the mechanisms. How does high ILT affect BDNF, serotonin, and depression? So that's a big question.
How does high IL-10?
Yes, IL-10.
That's a good question, and I don't know if I don't know if I can answer that question about IL-10 specifically. And I've been thinking a lot about this IL-10 finding because it's kind of it's definitely unexpected. And I think, what happens in the inflammatory cascade is that, your body produces pro-inflammatory cytokines and then begins to activate IL-10 to produce IL-10 to then help to stop the inflammatory cascade. And so part of me wonders whether the IL-10 finding is really just a reflection of this systemic inflammation that we are picking up on in the sample in the high-risk kids, rather than being an IL-10 specific mechanism. But I'm not sure, like it was such a unexpected finding that I wouldn't want to interpret it too much until I saw it in a few other data sets. But to me, it seems more plausible that IL-10 is just a proxy for kind of low-grade elevations in a number of different pro-inflammatory cytokines that IL-10 kind of followed to downregulate. But we're still kind of working through that.
Great. So thank you again for this great talk, and thank you for everyone in the audience and for your questions. And next week, our speaker will be Dr. Kim Revitz, and he will give the talk Gut Food Craving, How Gut Signals Control Appetite and Metabolism. So we will now move to Zoom for a Zoom question and answer. So if you're interested in joining, you can follow the link in the chat. So great. So thank you to everyone. And see you again. Okay.